Have you ever chosen the question with the fewest parts to answer, the most athletic kid for your PE team, or even the best looking watermelon out of the crate? What you're really doing is picking the most advantageous traits, which we have been doing for years to maximize benefits from the world. And mutations help us discover and select beneficial traits. To understand this, we first need to understand the basics of DNA. Traits are gene expressions where genes are segments of DNA containing instructions to produce proteins. DNA is a sequence of nucleotides. A codon, which is a sequence of 3 nucleotides, encodes a specific amino acid. Protein folding is when the polypeptide chain, a long chain of amino acids, folds into a functional 3D shape, guided by the amino acid's unique properties. Think of it like origami. The amino acids are the folds, the sequence is the order of the folds, and the protein structure and function are the final paper animal or object. Mutations are changes in the DNA sequence that can alter a protein's structure and function. Mutations come in various types, each with different effects. Substitution mutations include transitions, where a purine replaces a purine or a pyrimidine replaces a pyrimidine, and transversions, where a purine replaces a pyrimidine, or vice versa. Another category of mutations involve changes to a single nucleotide base, known as point mutations. These include silent mutations, where the base change does not affect the encoded amino acid and has no impact on the protein. Missense mutations occur when the base change leads to a different amino acid, potentially altering the protein's function. And nonsense mutations create a stop codon, prematurely halting protein synthesis. Frameshift mutations occur when nucleotides are inserted or deleted, shifting the reading frame and altering codon grouping. Induced mutagenesis intentionally creates DNA mutations that alter protein function. It helps scientists study impact of DNA changes on protein activity. There are 2 major types of mutagenesis. Site-directed mutagenesis is a PCR-based method that introduces mutations at specific DNA locations by targeting specific nucleotides or amino acids. Random mutagenesis introduces changes to DNA with less control over the modifications. It includes physical mutagenesis using agents like IR, X-ray, or UV to damage DNA, and chemical mutagenesis, which uses chemicals to alter DNA structure. This approach is useful for generating large mutant libraries to identify variants with improved or new unknown functions. Mutagenesis can introduce multiple mutations, but then we must account for epistasis, where mutations interact and affect each other. Mutations have shaped our biological world as we know it today and continue to drive innovations in medicine, agriculture, and sustainability. However, not all mutations are beneficial, so remember to wear sunscreen. Thank you for watching.